All right, so up to this point, we have talked a lot about molecular biology. We have talked about one specific class of molecules, proteins, and their building blocks, amino acids. We've talked about different families of proteins. We've talked about ECM proteins. We've talked about G proteins. We've talked about cytoskeleton proteins, and we've talked about in the last two lectures protein biochemistry and how we can leverage the biochemistry of proteins to analyze them, to quantify them, as well as how that's leveraged clinically as well. Today is going to be a pivot point where we start to talk about cellular biology, and we're going to be focusing on immunology. There's two arms of our immune system, and so that's how these lectures are broken up, where we're first going to talk about one arm of the immune system called the innate immune system or innate immunology, and we'll be talking about that in the context of Crohn's disease. So Crohn's disease is a type of inflammatory bowel disease. You may have heard of inflammatory bowel disease. It's an umbrella term that is meant to categorize specific diseases that deal with hyperinflammation of the GI tract. There's two diseases that fall under the category of inflammatory bowel disease. We have Crohn's disease and we have ulcerative colitis. As the name implies, ulcerative colitis means whenever there's itis on the end, that usually means inflammation of that tissue. So colitis is inflammation of the colon. So there's a few key differences between ulcerative colitis and Crohn's disease. Crohn's disease affects the entire GI tract, whereas ulcerative colitis is typically restricted to the colon. There's going to be lesions that typically emerge within both of them, but those lesions are slightly different between Crohn's disease and ulcerative colitis. We typically have in ulcerative colitis just the mucosal superficial layer that's affected, whereas with Crohn's disease, it can be across the entire wall of the GI tract there. And there's one last difference is what immune cells are attributed or associated with these diseases. They're adaptive immune cells, so we won't have the knowledge quite yet to really appreciate that cellular difference yet, but we can revisit that after we talk about adaptive immunology. But some of the symptoms associated with Crohn's disease is inhibited growth, fever, and weight loss. Because Crohn's disease can affect the entire GI tract, as some of you may have learned in physiology, the nutrient uptake within the small intestine is really crucial for your nutrition. And so the lesions that can impact or affect the small intestines can affect nutrient uptake, which then affect the patient's sustenance as well as their ability to grow and get what they need. There are across the GI tract these lesions that what can be described as, bless you, as abscesses. Abscesses are these infected regions that are pus-filled, and usually that pus is full of dead cells of the immune system, innate immune system, particularly neutrophils. And neutrophils are one of the cells of our innate immune system that are one of the first cells that arrive at a site of inflammation or infection. They're fairly short-lived. They come into the site of infection or inflammation, will release a lot of enzymes, can release a lot of proteins quickly, but then undergo death fairly quickly as well. And so that's why whenever there's infection, there's typically pus, all of these dead neutrophils that are there, and that's what an abscess is. It's also associated with pain, discomfort, diarrhea, ulcers, and in some more extreme cases, fistulas. A fistula is a connecting gap between two body cavities. And so two of the most common fistulas that show up within Crohn's disease is a fistula or a hole between the GI tract and the rectum or the GI tract and the vagina. And so these fistulas can have really deleterious, it can lead to really deleterious outcomes in patients with Crohn's disease. As you can imagine, because of the issues with infection, it can lead to anemia as well as some neurological issues as well. The prevalence. So there's currently about 1.5 million people that live with IBD in the United States. And so again, because it's IBD, it's both ulcerative colitis and Crohn's disease. When it comes to the prevalence, this is a little outdated, but currently it's about 1 to 10 cases per 100,000 people. And so that's actually on the rise over the course of the last 5 years. And so there's still efforts to understand the etiology of the disease to figure out why that is the case. And there appears to be a bimodal distribution among patients. Bimodal meaning there's, if you were to create a graph of numbers of patients across age, there seems to be two peaks, one at one within the teen years and one in, around 50. Again, there's not really a clear reason why. And so there's still a lot of work to be done to understand what's contributing to the age-associated presentation of Crohn's disease. So for etiology, one of the currently known causes is that there are genetic mutations that seem to predispose one or increase their susceptibility to Crohn's disease, and those genetic mutations are typically restricted to genes with a family of genes that are connected to innate immunology or our innate immune system. And what ends up happening is that when these mutations occur that affect our innate immune system, that affects their ability to function well, there's this overcompensation of the other arm of our immune system, the adaptive side. And so what ends up happening is this adaptive side overcompensates and becomes and contributes to a lot more inflammation that leads to Crohn's disease. And so a lot of the symptoms that we see among Crohn's disease patients is a result of this overcompensation of the adaptive immune system, and potentially as well as tied to microbial infections. And so our gut is filled with different bacterial species. And so there's this belief that it's a combination of genetic mutations as well as perhaps issues with the microbiome among some patient populations as well. One specific gene that is connected to or increases the susceptibility of Crohn's disease is NOD2. And so it's a gene that is connected with our innate immune system and has been identified to be connected with Crohn's disease as well. And so we're going to use that as our jumping point, and we'll revisit NOD2 towards the end of the lecture. But before we move on, I just want to give space for a couple of questions about Crohn's disease, innate immunology, or anything we've touched on so far. All right, we can press on. So our innate immune system, our immune system, every single immune cell originates from the same stem cell. They all originate from hematopoietic stem cells that are in our bone marrow. And so whenever there are instances of patients with some sort of genetic disorder that affects innate immune cells and their function, sometimes there can be bone marrow transplants that can help reverse that disease. And so that is meant to just try and highlight and indicate how all of our immune cells originate from hematopoietic stem cells. There's two cells that kind of emerge from the hematopoietic stem cell. So bless you. So this entire process, this lineage of immune cells and where they originate from, this is called hematopoiesis. For grad students in an immunology lab, like, it is expected of them to memorize this entire thing. You get familiar with it the more we stare at it, but I'm not asking you guys to memorize hematopoiesis here, but rather to try and help you see kind of the difference of our two arms of the immune system. So from the hematopoietic stem cell, you get a common myeloid progenitor and a common lymphoid progenitor. Everything that originates from the common lymphoid progenitor is the adaptive immune system. That's what we'll talk about next class. Everything that originates from the common myeloid progenitor is our innate immune system. The only exceptions here are erythrocytes or red blood cells, as well as megakaryocytes. But everything else that originates from a common myeloid progenitor constitutes the cells of our innate immune system. What I should also say is I updated the slides. I should have said this. This should have been my third announcement. I updated the slides to include a few slides like this. So I apologize if that ruins your note taking, but it's only 3 slides, so it's not a dramatic change. But this is just one of them to help illustrate some background. So we have our myeloid lineage, that's our innate immune system, and the cells that are indicated here are some of the main cells that constitute our cells of the innate immune system. So the important qualities of our entire immune system, there's 3 main qualities here that are true of both arms of our immune system, of both innate and adaptive. However, the degree to which they are true will differ for each. So our immune system is able to respond to a wide variety of pathogens. And so that helps imbue it with this ability to respond to a diversity of pathogens. Excuse me, pathogens being foreign microbes that our body interacts with or comes in contact with. Our immune system, I've said this a few times, but it is particularly noteworthy to try and emphasize this here. Our immune system is tasked with distinguishing between foreign versus self. It wants to promote tolerance and acceptance for what is identified as self and respond to what is foreign. Whatever is identified as foreign, whatever that molecule is, we term it as a pathogen. Our immune system is also incredibly specific. Our immune system, this is particularly true for our adaptive side, but true for both, where it's able to respond to very specific features or molecules that are on a pathogen, and not to pathogens that lack that feature. And so there's going to be multiple instances throughout this lecture where I'll be, for the sake of the recording, I'll say a virtual asterisk, for lack of a better phrase, where what is true of our biology will be distinct from the biology of pathogens, say viruses or bacteria. And so our immune system is able to distinguish on a very small molecular level what are features that are distinguished between our biology and the biology of viruses and bacteria. And so that gives it the specificity. And what's important to note is that our immune system has an incredible memory where they're able to respond to have a quicker and more readily available response to pathogens that were previously seen. This is particularly true for our adaptive side, but it's also true to a degree for our innate immune system. The idea of innate memory, I'll describe that in a little bit more detail, but this is also an entire field of research that's ongoing now. And so what I should note is everything we talk about in this class, I think this was an introductory lecture, it's worth repeating again. This is a class of half-truths. There are things that we know that are established, and there are things that have not been characterized and identified. This is particularly true for immunology. There are things that have been identified and characterized back to 1898, and there are fundamental things that are still being explored and characterized now. And so one of those things is memory, where there are things we know about memory, but there's a lot we're still trying to understand. So our immune system, again, is divided into two arms. We have the innate immune system and the adaptive. If you go back to that hematopoiesis slide, that is illustrated by where our immune cells originate from. But there's also a way to dissect our immune system into what are called cell-mediated parts and humoral parts. Humoral just meaning typically like bodily fluids, largely the blood, but it's also true for different bodily fluids, such as cerebrospinal fluid, peritoneal fluid, breast milk, lymphatics. And so it kind of constitutes all bodily fluids, but is particularly rings true for blood. And so under the innate immune system, the humoral immune response that we're going to talk about there is something that's called complement. We're going to go into detail and break down what the complement system is, why it's important, how it works. There's also cell-mediated responses within innate immunity. That's going to be largely, it's a little reductive, but largely phagocytosis. We've talked about that a little bit already, but phagocytosis, phago meaning eating, cytosis meaning by cells. And so phagocytosis is innate immune cells that are engulfing or eating foreign pathogens. On the adaptive side, the humoral response there is mediated by antibodies. We talked about last class how there is one specific immune cell that is tasked with making antibodies. That's B cells. So antibody production is the humoral part of adaptive immunity. The cell-mediated part is mediated by the cell-mediated immunity within adaptive immunity is permitted by cells actively eliminating or killing infected host cells of our own that are infected with something that need to be eliminated. So that could be, for example, virally infected cells. That could be tumor cells. There are cells on the adaptive side that clear virally infected cells, in some cases also tumor cells as well. So before we move any further, I want to give space to a couple of questions here because we're gonna jump in head first to these different parts of innate immunity. So again, to kind of like take a step back and give you the big picture, um, our immune system is broken up into innate and adaptive. We'll talk about adaptive next class. We're going to be breaking down both of those immune systems, both innate and adaptive, into a humoral part, what's in the bodily fluid, as well as a cell-mediated part. So what we're going to focus on next is the humoral innate immunity that is permitted by complement system. So the complement system is this family of proteins that's main task is to try and eliminate some sort of pathogen. These are proteins that are soluble, that are within our blood, and try and interact with different proteins to try and lead to the main function of complement is to try and promote the elimination of some pathogen that's been identified. There's three pathways for the complement system. We have the classical, we have the lectin pathway, and we have the alternative pathway. I'm going to first start with the classical pathway. What's important to note is what initiates or leads to the beginning of each of these pathways. For the classical pathway, what's going to end up happening first is that we're going to have, this is what's depicted in panel A in this image here. What you can see in the box is what is like the initiating event. What initiates this is we have some sort of antibody. We have some antibody that binds to the surface of some pathogen. Okay. All of the complement proteins, it's a bit of an alphanumeric soup. The alphabet part they all start with the letter C, which makes it easy to remember what the terminology is. And then the way they function is going to be in a numerical order. So we have C1 that binds to this antibody that's on the surface of the pathogen. That's going to lead to the recruitment of C2 and C4 that are cleaved into C2A and C2A, C2B, and C4A and C4B. Again, the takeaway is antibodies initiate this process, and they lead to downstream events that cleave proteins that, again, this is an important part. What ends up happening is that this C2A and C4B act on the C3 protein. C3 is an important like pivot point in this process. C2A and C4B is what we call a C3 convertase. That means they just cleave C3 into C3A and C3B. Okay. What ends up happening is that C3B stays there on the surface of that pathogen. C3A it leaves and it actually becomes an anaphylatoxin, which can recruit immune cells to this pathogen. Okay. So a key like pivot point, a key crux point is the formation of this C3 convertase in the classical pathway that is C2A and C4B. They cleave C3 into C3A and C3B. C3B stays, C3A leaves, it's an anaphylatoxin, it recruits other immune cells. And so we're going to push pause there on the classical pathway. That was oddly timed. I didn't mean for that to happen. The next pathway is the lectin pathway. So what ends up happening is the initiating event is that we have on the surface of some bacteria that is not on the surface of our cells is mannose. You typically won't find mannose on the surface of our cells. And so we have what's called mannose binding lectin, MBL, that will bind to mannose on the surface of bacteria. I'm going to step away here. And you can kind of appreciate that both mannose binding lectin and C1q kind of look almost like a spaceship docking on the surface here. And so like visually and functionally, they both kind of serve the same role here. But again, focusing on the key takeaways, antibodies initiate the classical pathway. Mannose binding lectin initiates the lectin pathway. And so we have mannose binding lectin that comes in. That's going to lead to C2 and C4 being cleaved. We have C2A and C4B, which again is a C3 convertase. It's important to know what the C3 convertase is. Its main function is to cleave C3 into C3A and C3B. C3B stays, C3A leaves, and it becomes an aflatoxin that recruits other immune cells to this pathogen. I'm going to push pause there. Okay, so we have our classical pathway that can be antibody mediated. We have our lectin pathway that's mediated by mannose binding lectin on the surface of bacteria. The alternative pathway is happening is the last final pathway that's possible here. So what ends up happening sometimes is that C3 can actually be via hydrolysis cleaved into C3b. There's a couple of other soluble factors in the blood, factor B and factor D. When they, again, I'm gonna step away from the computer here. When C3b binds to these two factors, we have the formation of another C3 convertase. So the C3 convertase in the alternative pathway is C3b with these two factors that lead to the cleaving of C3 and production of C3b on the surface there. So what can happen is that while we have the hydrolysis of C3 that happens kind of spontaneously at times, what we can have is that the C3b produced by the classical pathway can feed into the alternative pathway, and you kind of have this amplification of complement that feeds forward and accelerates the response to this pathogen. But the key thing to note here is that all three pathways converge at forming a C3 convertase. Okay? There's a formation of this enzyme that cleaves C3 into C3A and C3B. What's a little nice about complement, despite it being a large alphanumeric soup, which, just to warn you, is true of all of immunology, which is why I love it. That was a joke. That was a bad joke, forgive me. Despite it being an alphanumeric soup here, each protein, when it's cleaved, turns into its these A and B counterparts. And so while it seems a little intimidating to read this and to hear this, there's a bit of a pattern to how complement works, where it's happening in numerical order. Each protein, when it's cleaved, falls into these A and B constitutive parts here. And despite perhaps feeling like you're drinking from a fire hose with this alphanumeric soup, the key takeaways here is that complement is the humoral immune response in innate immunity. There's three pathways that are involved with it. The classical pathway initiates with an antibody binding to the surface of this pathogen. Mannose binding lectin, the lectin pathway initiates with this mannose binding lectin molecule that binds to the mannose on the surface. Excuse me. We have the alternative pathway that's a bit of a feed-forward loop, but all three converge at the formation of a C3 convertase that promotes the formation of C3b that is in the surface of this pathogen, while C3a leaves and is and promotes the recruitment of other immune cells. Now, what can end up happening here is once we have C3b there, we'll actually have the formation of a C5 convertase. Oh, sorry, let me take a step back. C3b on the surface can actually serve as a signal to phagocytes to eat and destroy this pathogen. We have the formation of a C5 convertase after C3b is there. That leads to the cleaving of C5 into C5a and C5b. C5a is also an aflatoxin. So there's the recruitment of immune cells by these aflatoxins, C3a and C5a. We have C3b in the surface that serves as kind of like a signal for phagocytes that are being recruited to now come and eat this pathogen. Now, if that doesn't completely destroy the pathogen, the completion of complement is what's the formation of what's called the membrane attack complex. What ends up happening is after we have the C5 convertase that cleaves C5a and C5b, C5b is wedged in the surface, C5a is an aflatoxin that's released. Then we have C6, C7, C8 that comes in. And after that, we have C9 that creates almost drilling a well in the surface of this pathogen that is happening all over the surface that it will now destroy and eliminate this pathogen. So the completion of complement is the formation of the membrane attack complex, or MAC, where it's perforating the membrane of this pathogen and leading to its destruction. I'm going to step back and give space for a few questions here. Yes. When you say all of it, what do you mean? All of the different complement proteins. Great question. So we have C3b that when it contributes to the formation of the C5 convertase. Once we get C5b, that's what begins the formation of the membrane attack complex. So it's C5b to C9 that are part of the membrane attack complex. Does that help clarify? The precursors are not part of the membrane attack complex, but they help lead to the formation downstream of the membrane attack complex, but they're not physically a part of it. Yes. So we have once we have C3b coated on the surface of the pathogen, we have the C5 convertase. Let me see. Sorry. C3b interacts with a couple of different markers, a couple of different proteins that contribute to the formation of the C5 convertase. So C3b contributes to the formation of the C5 convertase. And once you get the C5 convertase, now you get C5a and C5b. C5a is an aflatoxin, it's soluble, it leaves and recruits immune cells. C5b is wedged in the surface. So when it comes to like trying to figure out what some of the patterns, C3b and C5b are wedged in the surface of the pathogen. C3a and C5a lead. They're aflatoxins, they're soluble factors, they're in the blood, and they're going to be trying to recruit immune cells to this infection. Yes. Yeah.
Complex, is that what all the students have, or is that all the complex?
Say that one more time.
In the diagram.
Yes.
Is
Yeah.
that like all made up of? Or?
Yeah, it's all made up of that.
Okay. And
Yeah.
do those have like different things or specific structures?
Are there different receptors for those complement proteins? Is that what you're asking? Not for C6, 7, or 8. There are receptors for C3a and C3b. Sorry. There are receptors for C3a and C5a because they recruit immune cells to this site. So they'll bind to the surface on the receptor and recruit cells here. There's also receptors for C3b. C5b, I don't believe cells express a receptor for that. Yeah, I'll stay there. I'm pretty sure there's not receptors for C5b. I could be wrong. I don't think there are. Does that answer your question?
Yeah, so is there anything binding to that?
Great question. There's nothing binding to this complex. I'm going to step back for the sake of the recording. The main function of the membrane attack complex is to perforate or poke holes in this pathogen to lead to its destruction, to eliminate it. Yes.
For the data set mentioned, are those part of the
They are. So this is a great question. So typically, the humoral response can lead to the cell-mediated response. So in cases where perhaps while the membrane attack complex is forming, what can happen is that a cell-mediated response is elicited. And so that's kind of one way in which these are connected, if that makes sense. Does that answer your question? Both pathways being humoral and cell-mediated, or in a way, yes, but what I want you to focus on is largely that's 100% true. And in biology, yes, like humoral leads to cell mediated response. That's true in innate and that's true in adaptive. They're not completely divorced from one another, but they kind of work in concert with each other. Yeah, go ahead.
Instead of like involving antibodies,
Mhm.
is that more of like the adaptive immune system, or would that have to do things?
That's a great question. So no, it's a great question. The antibodies originate from the adaptive immune system. The presumption here is that these antibodies are already circulating in your blood. So we're not relying on the B cells in our adaptive immunity, our adaptive immune system to generate new antibodies, but rather relying on pre-existing antibodies that are just already in your bloodstream circulating that recognize this pathogen. Does that help? I'm gonna go with the last question here. We're gonna move on. Yeah, so the Manos is actually highlighted in with that blue box on the right side. Um, sorry? Yeah, so these carbohydrates are Manos. That's the species of carbohydrates they are. And so we have this lectin, that's why it's called the blue spaceship looking structure, is lectin. That's why it's called the lectin pathway. Specifically, it's a Manos binding lectin, which is why it's labeled as MBL up top here. Great questions. I want to keep answering them, but we got to press forward. My goal is to try and finish this today. We'll see if we get there. So as we've talked about already a little bit, there are molecular features that are present on a lot of pathogens. And we have receptors that recognize these different molecular features. When these features are associated on a specific pathogen, they're called pathogen-associated molecular patterns, or PAMPs. And what recognizes PAMPs are pattern recognition receptors, or PRRs. Excuse me. So pattern recognition receptors recognize PAMPs. Again, PAMPs are just these features on pathogens that are unique to pathogens. Again, virtual asterisk. They recognize what is distinct from pathogens from our biology. And so this has been, these PAMPs have been, over the course of evolution, have been distinguished and have led to the formation of pattern recognition receptors that contributes to this long-term memory of our innate immune system. When these pattern recognition receptors, again, the memory here is that they are encoded in our DNA and recognize PAMPs. And when a pattern recognition receptor binds to and recognizes a PAMP, it triggers certain outcomes from innate immune cells. What it can lead to is chemotaxis. What that is the movement or migration towards of a cell towards that PAMP. And so whenever you see taxis in some scientific term, think movement or migration of that cell. So PAMPs can lead to chemotaxis, movement of cells towards that PAMP. We can also benefit from defensive responses that attack that pathogen. We also get the release of these proteins that are signaling proteins between cells that are called cytokines. They lead to the activation of cells. They lead to their growth, their expansion. But cytokines, if you just want to categorize it broadly, are just signaling proteins between cells, particularly immune cells, that lead to their activation. They also lead to the release of chemokines. Chemokines are proteins that promote chemotaxis. And so they lead to the migration and movement of cells towards that pathogen. And finally, the binding of PAMPs on a pattern recognition receptors can lead to changes in gene expression where certain genes are activated that lead to downstream activation of a cell. We're going to talk about a few different kinds of pattern recognition receptors. Again, we're going to focus on the big picture of what do these pattern recognition receptors bind to, and what's how does that distinguish from our own biology from the pathogen? So one is called formal peptide receptor, or FPR. It's a class of G protein coupled receptors that recognize formulated peptides that are uniquely on bacteria. So the reason why it's unique to bacteria, what ends up happening is that every protein in bacteria and in human biology, the first amino acid is always methionine. Once we get to the next unit, you'll be able to appreciate why that's the case. But what distinguishes our proteins from those of bacteria is that their methionine is formulated. Ours is not. Therefore, over the course of evolution, our immune systems have developed a formal peptide receptor, a receptor that recognizes peptides that have that are formulated on that methionine. And so binding of the FPR is a really potent chemoattractant, meaning it promotes the movement of cells towards that pathogen, particularly in neutrophils and macrophages. What's worth noting is that in our genome, we encode for 3 kinds of FPRs, whereas mice actually have 6. Because we are a higher order organism than mice, typically our genome has more copies of certain genes. However, this is one instance where mice actually have more diversity than we do when it comes to encoding for the FPR. The main reason for that is if you think about what conditions mice live in, they are exposed to far more bacteria than we are. So it is evolutionarily advantageous for mice to have more receptors that recognize and attack bacteria because they're simply exposed to a lot more than we are. At least they should be, by and large. And so this is the formulated group on that methionine amino acid here. Another family of pattern recognition receptors, the most well-characterized, most well-known, are toll-like receptors, or TLRs. They're called toll-like because they resemble a family of genes that were originally discovered in flies, toll receptors that were in flies. And so because they resemble those receptors, they were just called toll-like. It's a fairly unimpressive nomenclature, but they are probably some of the most important and most well-characterized pattern recognition receptors in innate immunity. Some are localized on the cell surface. Some are localized within the cell, and that depends on what they bind to. We have about 10 that have been identified. I'm just going to narrow in on just a few of them to draw your attention to. And again, it's worth noting that our toll-like receptors recognize molecular features that are unique to pathogens that are not present in us. So for example, double-stranded RNA is recognized by TLR3. Our RNA is single-stranded. So there should not be single-stranded RNA within our cells. If that is the case, it's from some sort of virus here. Flagellin, which is found on bacteria. LPS is lipopolysaccharide. It's also called endotoxin. It is an incredibly potent innate immune activator that binds to TLR4 and TLR2 a little bit, but it is, again, keeping things simple, just think of LPS as a TLR4 ligand. It binds to TLR4. We also have LTA, which is lipoic acid. And finally, we have what's called CPG. That's our nucleotides, cytosine and guanine. And what's worth noting is that typically, not universally, but typically, when we have cytosine and guanine next to each other in our DNA, they're usually methylated. There's a methyl group added. That is not the case in bacteria. So that's another way in which our biology is different than bacterial biology. And so these, and I'll provide a little bit more information about 2 of these TLR ligands, LPS and LTA in a moment. But to help visualize what this looks like, the toll-like receptors that would bind to their ligand or encounter them extracellularly are on our cell surface. The TLRs that would encounter their ligand when that pathogen is engulfed, those are actually present on the membrane of our endosomes. And so their location is totally informed by what they bind to and what they recognize. Lipopolysaccharide, or LPS. So that is a what it has is it is a marker that is on the surface of the bacterial cell wall that is present on specifically gram-negative bacteria. Again, it's also referred to as endotoxin. So whenever you come across something that is where you hear endotoxin, think LPS, think that it binds to TLR4 and that it's a potent activator of innate immune cells. The next PAMP, so I should say LPS and LTA, they are these are patterns, molecular features on the surface of some classes of bacteria. Lipoteichoic acid is also on the surface of bacteria, found specifically on gram-positive bacteria. The next slide, I'll show you why that's the case. But it is another potent activator of TLRs here. And the reason why they're on gram-negative versus gram-positive bacteria is because on gram is a stain that was identified really early in immunology to try and distinguish between different classes of bacteria. The gram stain binds to peptidoglycan. So the species of bacteria that outside their membrane where peptidoglycan is accessible, the gram stain will be retained by this peptidoglycan and stain positive, gram positive. For gram-negative bacteria, again, that's lipoteichoic acid. We go back over here. On the right half, for gram-negative bacteria, there are two membranes. There we go. For gram-negative bacteria, there are two membranes where within it is the peptidoglycan. So it's not accessible to the stain. It's going to stain negative. That's why it's called gram-negative bacteria. And that's going to be what LPS is going to be on the surface of. And you can see that at the top on the right half here of the gram-negative, where there's LPS, lipopolysaccharide, on the surface of that outer membrane. I think second to last, almost last, again, is cytosine phosphate guanine, or cytosine guanine. Again, it's these two nucleotide bases where, again, in our biology, typically what you'll see is methylated CpGs. In bacteria, they are not methylated. And so we have a specific TLR, I believe it's TLR9, if I remember correctly, that recognizes and binds to this PAMP. It's worth noting that, again, I described earlier how these things are always being better characterized and better identified. A professor here at UVA was part of the research group that determined and characterized the toll-like receptor TLR9 that binds to this PAMP. And so, again, this is an ongoing active area of research by and large here. So CpG binds to TLR9. Again, it's usually methylated in our biology, is unmethylated in bacteria, and that's how we can distinguish between us. And that's where the methyl group is added on the cytosine there. This is the second to last, is C-gas. So C-gas senses cytoplasmic DNA. Again, our DNA is typically within our nucleus. If there's DNA in the cytoplasm, that's a problem. It's coming from some sort of pathogen. So what ends up happening is that when C-gas binds to and senses DNA within the cytoplasm, it generates cyclic GAMP. What ends up happening there is that then cyclic GAMP activates a molecule called STING. STING leads to the expression of interferon. So again, the key thing to note here is that C-gas recognizes and senses cytoplasmic DNA. DNA shouldn't be in the cytoplasm there. These downstream events lead to the expression of interferon. That's a protein that prompts every cell in your body to express a specific special protein. When it comes to organ transplantation, you guys have heard of the idea of tissue matching. There's a specific protein that is unique to you and only you. That is almost like an ID card for your cells. In the case of cytoplasmic DNA being present, that indicates that perhaps there's a virus that is within that infected cell. So when interferon is expressed, every cell expresses that ID card protein. They express it way more to indicate to our immune system, I am who I am and I'm supposed to be here. But cells that are infected, that have cytoplasmic DNA, express that ID card protein much less. And so then that helps us identify who is the infected cell and who needs to be eliminated. Yes.
Cancer, this was something happening with, and you got infection on top of that, and then your body started expressing protein. With the cancer cells, also you just like recognize the expressing protein.
That's a great question. That the combination of viral infection during cancer is an ongoing area of research. So I cancer biology is not my domain. So I don't know, but I do know that is a current active area of research. Yeah. The last one that I'm going to talk about that's going to wrap things up, and I'm going to do it in like a minute and a half, but not rushed where it's like you're overwhelmed and drinking from a fire hose. It's going to be like refined and elegant minute and a half. The last pattern recognition receptor that we're touching on today is NOD2. It recognizes, again, peptidoglycan. We talked about how that's what the gram stain binds to. And so it recognizes peptidoglycan on the surface of a bacteria. Specifically, it binds to what's called muramyl dipeptide. What that is, let me go back over here, forgive me. Muramic acid with two amino acids down here. And so this is N-acetyl muramic acid. That's this species of muramyl dipeptide. NOD2 is expressed in some of our phagocytes, specifically monocytes and macrophages, as well as also in gut epithelia, which is our connection point back to Crohn's disease. So what has been, again, I'm going to say, again, NOD2 is a pattern recognition receptor. The PAMP that it recognizes is muramyl dipeptide. So our connection point is that NOD2 has been identified as a susceptibility gene for Crohn's disease. So the three biggest journals in the world are Nature, Cell Science. If you ever publish in one of those journals, that is a huge accomplishment in your career. In Nature, back in 2001, there were two back-to-back articles that looked at susceptibility genes and NOD2 that identified that there was a linkage analysis looking at genes that are affected that are connected to this disease of Crohn's disease. And what they identified is that there's something that happens, there's a mutation on chromosome 16 that seems to increase the susceptibility of one for Crohn's disease. The very next article after that identified that NOD2 specifically seems to have a mutation that is associated with Crohn's disease. Interestingly enough, NOD2 is on chromosome 16. What they identified through a familial analysis, 1 in 4 are the parents, 2 in 3 are their children, is that in this family, each parent had the wild type allele for NOD2, the normal allele, and a shortened version of NOD2. Both children inherited that truncated form. Both children had Crohn's disease. So what ended up happening in a follow-up study is, okay, a group took that truncated form of NOD2 and a genetically engineered mouse replaced NOD2 with that truncated form and looked to see what happens to the GI tract. What ended up happening was that in a way of inducing colitis, in this case it was colitis, there's not a great model for Crohn's disease, but there was a much more severe inflammation of the GI tract. You can appreciate here that the GI tract is just much thicker and larger compared to here, as well as if you zoom in, you can see that there's a much thicker GI wall of indicative of inflammation that indicates that this mutation that was identified in this family, when you genetically engineer a mouse and put that mutation in, it leads to inflammatory bowel disease. So for treatments for Crohn's disease, typically patients will take corticosteroids to try and reduce the inflammation. Specifically, they'll take antibodies that neutralize and get rid of tumor necrosis factor, or TNF, which is currently Remicade and Humira. Humira, I think, is the second most widely used drug in the market right now. And so it's not a cure-all, but helps to relieve and alleviate symptoms. If innate immunity or Crohn's disease interests you, there's a great group of folks here at UVA, as well as 2 folks at other universities whose work I really appreciate. I wanted to give space for more questions. I tried to stick to my minute and a half. I didn't, forgive me. Thanks for your time, and I'll end it there.